nervous system. Give me some ideas. Anybody just popcorn out some ideas about the nervous system. What does it do? What does the nervous system do?
Reacts to outside.
Reacts to outside stimulus, right? Stimuli is the word we'll use. What else? So yes, it's picking up outside signals, and your body's responding to those. What else is the nervous system doing? In addition to that, it's responding to outside stimuli, and I guess the word respond, right? You're going to take action, perhaps, right? Your nervous system may respond by having you move a muscle, or maybe by secreting something, maybe by controlling your heart, or controlling your digestive system, or a reflex like blinking, or moving away from a hot stove. So your nervous system is coordinating, right? All the marvelous things that we do, and it does so in a way that I think you'll be in awe of. We'll spend a lot of time next semester in 106 talking about exactly how this works. This semester, we're really focusing more on the structures involved in the nervous system, learning a little bit about the heart, I'm already ahead to the heart. Learning about the brain, learning about nerves, and about the reflex arc. We'll learn about the eye and the ear. Anatomically, we can divide the nervous system up into two different divisions. This is very, very straightforward. There is the central nervous system, and that is, as the name suggests, right in the center. That's going to be your brain and your spinal cord. So the brain and the spinal cord are your central nervous system and the things included within the brain and spinal cord. And then there's the peripheral nervous system, and as the name suggests, everything in the periphery. Everything outside of the CNS is part of the PNS. This is going to include the nerves of your body, and this term ganglion, which you're going to hear over and over and over. It means nothing to you yet, but a ganglion is a bunch of cell bodies, a bunch of neuron cell bodies that are clustered together, and we'll be talking about ganglia quite a bit. Ganglion is singular, ganglia is plural. Let me see that word for you. Okay, so ganglia plural for ganglion. Now, looking at this in a picture, this drawing doesn't quite have the difference in color that I would like to have. Remember that yellow is sort of the universal color for the nervous system. Here we see a bunch of yellow nerves. All of those yellow nerves on this image are part of the peripheral nervous system. The central nervous system on this particular diagram is shown in a tan color. So that's the brain and the spinal cord. Okay. Everything else is peripheral. We'll come back to the brain. For now, let's talk about the spinal cord a little bit. Tell me about the spinal cord. What's your understanding of it? You did some pre-lab exercises on this, but what do you know about the spinal cord? Where does it run? Where is it found? Say it again? Along the back. In what specific space? We saw this in the very first lab when we turned the body to the side and we talked about the dorsal body cavities. There was a cranial cavity where the brain is found, and the spinal cord was housed within the vertebral canal. And the vertebral canal, we know, is actually traveling through what series of openings? The spinal cord is traveling through what? I told you you wouldn't be quizzed on bone markings, but I lied, okay? So what's that opening going through all of your vertebra through which the spinal cord passes? The vertebral foramina, right? Plural. All of those vertebral foramina are the opening for the spinal cord. And you learned in your pre-lab information that the spinal cord does not go all the way down to the sacrum or down to the coccyx. It goes how far? The spinal cord itself travels, it leaves the brain from the brain stem, travels through what opening in the occipital bone? Foramen magnum or magnum foramen, right? Passes through that opening, continues down through the vertebral canal and through the vertebra foramina until where? About L1. And so the spinal cord is going to travel down to about L1. L1, your first lumbar vertebra. The spinal cord itself has regions, and boy, these regions look really familiar to you. Cervical, thoracic, lumbar, and sacral regions. And this is where I'll back up and wait for a question. I'm just going to wait for a question. Hopefully not too long of a wait.
How was your break?
My break was fine, thank you. How about I frame the question this way? Dr. Macauley, you just said that the spinal cord only goes down to L1. So if the spinal cord only goes down to L1, how in the world is there a sacral region to the spinal cord? Is that a good question? Okay, now we see where I'm waiting for, okay? So the spinal cord only goes down to L1. That's the spinal cord proper, okay, the actual structural spinal cord. But there are these regions assigned, and in a moment I'll show you this picture. So I'm not going to answer the question quite yet, but I want you to be curious about this. Coming off from the spinal cord, there are 31 pairs of nerves. You saw these nerves when we were looking at the vertebral model earlier in the semester, or I'll grab this smaller one for now. What do you see? Okay. So on this model, what are these yellow structures? These yellow structures are the spinal nerves. Okay? And you notice that the spinal nerves are coming off from the spinal cord through what opening? I lied a third time. What's the opening? Intervertebral foramina, right? The opening between the vertebra, and you can see that these spinal nerves are coming out through that opening. Okay? So again, we can't forget everything, can we? So there's 31 pairs of these nerves. Let me give you some numbers right now so you understand where these 31 nerves are coming from. How many vertebrae are there? My fourth line in a row. You have to remember that too. How many vertebrae are there? Remember? We eat breakfast at 7, lunch at 12, and dinner at 5. So how many? 24 vertebrae, right? But I'm telling you there are 31 spinal nerves. So let's figure out where those 31 spinal nerves are coming from. So this is vertebra, and this is now spinal nerves. Cervical. There's basically one nerve per vertebra. However, in the cervical region, there is an 8th cervical nerve. Okay? And you'll see that in the models. So there are 8 cervical nerves. They're going to be numbered and named similarly to the vertebra. So they're going to be named C1, C2, C3, C4, all the way down to C8. This can be a little bit confusing. You'll look at the context to determine, are they talking about the atlas, the bone C1, or is the conversation referring to the nerve C1? Okay, so you'll see this double naming. So there are 8 cervical spinal nerves, and then the numbers after this kind of make sense. There are 12 thoracic, again 1 for every vertebra. There are 5 lumbar, L1, L2, L3, L4, L5. There are 5 sacral, 6th line in a row. Those nerves for the sacral region are passing through what openings? On the sacrum, what were the openings through which those sacral nerves pass? Dorsosacral foramina. Very good. Okay, so remember those openings in the sacrum. There are 5 nerves that are called sacral nerves. And then there's one coccygeal nerve. Okay, and sometimes that is abbreviated CO1 or just CO. And that's so it doesn't get confused with C for cervical. Okay, so we add that up together. What do we get? What do we get all together now? 8, 12, 5, 5, and 1? Better be 31, right? So we have 31 pairs of spinal nerves. So this does include the sacral nerves. That's different, right, than in the vertebra. We don't consider the sacrum a vertebra, do we? But now we include these nerves, the sacral nerves, as part of the spinal nerves. Also, coming off the spinal cord in 4 distinct places, there's a group of nerves. And you'll see this very, very clearly on some of the models. And this group of nerves is called a plexus. A plexus is a weaving, an intertwined group of nerves that travel together. And those 4 plexuses, I'll list them now. There's cervical, C, brachial, B, lumbar, and sacral. Okay? So cervical, brachial, lumbar, and sacral plexus. Brachial plexus. Where must that group of nerves be going? To the arm, right? To the brachium. Good. So we'll see how that works as we look at the models. And all my scribble up on the screen. We're also going to get to know the neuron very, very well. We've looked at cells before. We saw the typical spherical cell with the purple nucleus and the green Golgi that we labeled back in the first couple of chapters. We saw that muscle cells are very, very different. Remember, this muscle cell has the sarcolemma and has the myofibrils, and the muscle cell has its own architecture and its own terminology. Well, the neuron is also a very unique cell. It has a very different structure, and so therefore it also has its own vocabulary. We're going to learn about this cell in great detail over the next couple of weeks. Actually, today's lecture, tomorrow's lecture, is largely on the neuron and understanding the parts of the neuron and how this particular cell does some of its magic. So this is the prototypic cell that you'll be labeling, and you'll learn how to label this very, very well. Some of these can be the longest cells in your body. Remember I talked about how the muscle cells can be very long? And that muscle cells are as long as a muscle is. So like the sartorius, that's a pretty long muscle. Those cells can be 18 inches long or so. Neurons can be 3 feet long. Right? You can have a neuron that goes all the way from your spinal cord all the way down to your big toe. So in a person my height, easily a 3-foot long cell. So that's very different than what we've experienced before. The majority of the cell is referred to as the cell body. That's going to be this spherical region. Okay? We'll call it the cell body. Another name for that is the soma. Okay, so the cell body or the soma, same thing. You'll see them used interchangeably. Now what's unique about the neuron is that it has many processes sticking out of it or coming from it. What's a process? What was a process back in bone world? The spinal process, spinous process. What was a process? Something that stuck out, right? Something that protruded out. And so that's true here as well. So there are many processes coming out of or entering into the cell body. Many, not all, but many neurons have multiple little branches coming inward, and these are called dendrites. Now we know what dend means from our vocabulary. Dend means tree. Ite would suggest that these are little trees, right? So the dendrites are these little tree-like branches, and the information that travels through a neuron does so in essentially a unidirectional way. So the information, the electrical information, the stimulus is going to be coming in to the soma through the dendrites, moving toward, from the outside, toward the soma. This cell has a nucleus like any other cell does. This is a very metabolic cell. Lots of activity going on in this cell. It has a lot of mitochondria. It needs to. It's a very energy-needy cell. It has a very distinctive nucleolus. Remember what the nucleolus did? This is going back a couple of chapters. That's my 7th lie of the day. You do have to remember some of this stuff. What's the nucleolus do? It was that dark staining center of the nucleus. When you were thinking about your organelles, I told you to think about the one thing that each organelle does. And the nucleolus, or the nucleolus, pronounced both ways, was responsible for making ribosomal RNA or synthesizing the ribosome, something like that in memory bank. So this is a very, very highly transcribing, translating cell. So it has a very big nucleolus because it is very, very busy making ribosomes and making protein. We'll talk about what those proteins are later. Then, attached to the neuron cell body, there'll be one and only one much longer, more substantial process referred to as the axon. And you see this axon is much, much longer. The information flowing through this is shown to us in the red arrow there. It's telling us that information is flowing unidirectionally away from the soma. So axon away, AA. The information is flowing away from the soma down the axon. Now again, this axon could be very, very long. This is where the length of the cell comes from, is this axon. At the very end, even before then, it is possible for the axon to branch. There's a branch. Here's another branch. But you'll only see one axon actually attached to the soma itself. At the very, very end of the axon are some little branches. Okay, the axon will branch. If my head was the soma, right, and my arm is the axon coming off, then my fingers would be these little branches at the end of the axon. Those branches go by 3 different names. Okay, this particular diagram is calling them the terminal arborizations. So terminal suggests that it's at the end, right? At the very end of the axon. And what do we do on Arbor Day? On Arbor Day, one plants trees. So what does that kind of tell us? Terminal arborization is a really fancy way of saying there's little branches, little tree-like branches at the end. Not to be confused with dendrites. These are just the little ends of the axon. The easiest thing to remember for these, this is equal to axon terminal. Okay, so each of these is an axon terminal. Again, that's probably the easiest of the 3 names to remember. The third term to describe the same exact thing, I'll put the word up here, telodendria. Let's break that word apart, shall we? What does telo mean? End, right? Where have we seen that word before? In mitosis, right? Telo was the last phase of mitosis. So telo means the end or the last. And at the last or at the end, what do we have? Little trees. Okay? Not to be confused with dendrites again, but these little tree-like branches at the end of the axon. So these little terminal arborizations, these little axon terminals, these little telodendria, you will see all 3 words used, depending upon what book you're opening up, and depends on how I'm feeling that day as to which one I call it. At the very, very end of those axon terminals, there's a little swollen end, and that's referred to as the synaptic knob. Synaptic knob. What happens at the synaptic knob? Anybody know? It synapses, right? What's a synapse? I'm throwing a lot of things at you. There's a whole new vocabulary here, folks. The synapse. Let me clean this up a little bit. What does APS mean from our vocabulary? APS or APT? It was the very first list. Further evidence I should make vocabulary cumulative, right? APS or APT, to fit or to fasten. And then synthesis, symphysis, right, to come together. So a synapse is where 2 things are going to come together in very, very close proximity. And it's there, what's released there at the synapse? What is released at the axon terminus? Chemicals called neurotransmitters. Absolutely. Neurotransmitters. Remember I told you this cell was very, very busy making proteins. And this cell is having to constantly make neurotransmitters. These are mostly a group of proteins, and they're going to be made up in the soma and released at the axon terminus, or at the synaptic knob. Okay, and we'll talk more about this. Now, the other thing we see is that along the length of this cell, not the whole cell, but along the axon, I see that there's another cell that is wrapped around the axon. That cell is protecting the axon. Axons are very special cells. They're like a queen bee. They have to be well protected. Neurons cannot divide. Okay, that's something important to remember. Neurons do not have what organelle necessary for cell division? Spindle apparatus, they don't have centrioles. You're absolutely right. They don't have the centrioles. Remember what the centrioles were? They were those little structures from which the mitotic spindles kind of spit out like a Spider-Man, right, to grab the chromosomes. They don't have centrioles. So neurons can't divide. So once they're formed, that's it. And that's why brain damage and spinal cord damage is so devastating, because at this point, we've not figured out any way to trick neurons to divide. So once damaged, that's it, right? You sever the spinal cord, that's pretty much it. We don't have a great way of regenerating those cells. So we have to protect this cell during its entire length of your life. These cells along the axon are really helping to do a few things, but they're definitely protecting the axon. And these cells are called Schwann cells. Okay, Schwann cells. And these Schwann cells are making a very important protein. It's a fatty, white protein called myelin. And that myelin is protective, but it's also like electrical tape. It's going to help maintain the electrical signal as it travels down the axon. So more about myelin, more about Schwann cells later on. That's quite a bit of information already, isn't it? Just by looking at that picture. As you do your pre-lab, as you've done your pre-lab, go back and look at that. Also, as you listen through lecture 1 today and tomorrow, listen to all of these structures that I've said. One more thing, 2 more things while I'm here. In between the Schwann cells, you see a little break? There's a little tiny break in between each of these Schwann cells. That was described by a guy named Ranvier. So those little breaks are called the nodes of Ranvier. R-A-N-V-I-E-R, a French word. If you're in Texas, it's the Ranvier, right? But the nodes of Ranvier. And I think that's all I have to say about the neurons for now. But you will get to know and love this cell and know its parts well. Questions? Okay, so that's what a neuron looks like, but not all neurons do the same thing. Some neurons are sensory neurons. As we suggested at the beginning, that means that they're going to sense the environment. This environment could be in your external environment, right? So the temperature in the air right now, the sound of my voice, the light that you're seeing, those are all sensory signals that are being picked up by your sensory neurons. The other word for sensory is afferent. Now, I'm going to really overemphasize the word afferent. Most people will just say afferent, right? But I'm going to really overemphasize afferent and efferent as I discuss these for the next few weeks. What do we know about AF as a prefix? AF, that's my fault. AF means what? Toward. Ferent means to carry. So what does afferent mean? To carry toward. To carry toward what? We're going toward the center. We're going toward the brain and the spinal cord. We're going toward the CNS. So these are your sensory neurons. They can be located in 2 basic places. In your gut. Right now, your stomach is sending signals to your brain. You're not aware of it, but your kidneys and your stomach and your lungs and your heart are all sending signals to you from your viscera. Or these could be sensory neurons, afferent neurons found on your periphery. Your hearing, your vision, your touch, your smell. Those are all sensory neurons as well. I'm going to pop down to the bottom. Then there are motor neurons. As the name suggests, they're going to cause you to move or do something. The other word for these motor neurons are the efferent neurons. And EF meant what? E and EF, to exit, right? To leave, to go away from. So the efferent neurons are carrying signals away from the central nervous system, away from the brain, away from the spinal cord, and these are going to go to one of 3 places. They're going to go to your skeletal muscles, actually 4 places. They're going to go to your skeletal muscle, telling you to move. They're going to go to your smooth muscle, telling it to contract or relax. It's going to go to your heart muscle and help regulate your heart rhythm. And it's also, these motor neurons can also go to your glands, like your salivary glands or your lacrimal glands, your tear glands, and cause you to secrete something or not. Okay, so you've got movement or secretion related to these motor neurons. Now in the middle, there are some interneurons. What does inter mean? This is our last vocabulary list. This should be more fresh. Inter means between, right? So the interneurons are found what? Between the incoming sensory and the outgoing motor neurons. These interneurons are found wholly and completely within the CNS. So they're completely contained within the brain. They're completely contained within the spinal cord. They're the connectors. They're the processors. They're the retrievers. They're the integrators. They're the real, if you will, brains of the nervous system, and that they're receiving this information. Okay, so we know that neurons have a particular shape. We looked at those. We know that now neurons have different jobs. And a sensory neuron would only carry information in. And a motor neuron would only send information out. The information will be unidirectional. So, remember last semester I had a table that looked a lot like this about skeletal, smooth, and cardiac muscle, and I asked you to know its different features and functions. Same idea here. I'd like you to fill in for a moment, kind of take a break from listening to me, and fill this in. Sensory, motor, and interneurons, what do they do? Where are they located? And for now, leave this last column empty, their class. But go ahead and just fill it in briefly, and we'll come back and discuss that, and we'll put this all together. Let's see what you filled in for this. So let's start with the sensory neurons. What is their function? Don't make this hard. To detect, to receive, right? To detect and receive. And where are they located? These are sensing, right? These are your sensors, if you will. So where are we going to find? Two basic places where we're going to find these sensory neurons. Say it again. Yep, the viscera, right? In your gut, definitely your heart and your bladder right now are telling you things that you're not aware of necessarily, but certainly you are being told from your viscera, and so that'd be your internal environment, and your internal environment, but also a lot of your sensory neurons are located on your outside of your body, right? And they are picking up your external signals, aren't they? So we have internal and external sensory neurons. How about motor neurons? What do they do? I'm just going to say send. Yeah, they're going to send signals out. And where are those signals going to? They're going to end up going from the brain or the spinal cord, and they're going to go out to your muscles, or they're going to go out to your glands. All three kinds of muscles, right? Skeletal, cardiac, and smooth muscle. And out to your glands. And then the interneurons, their job was what? Store, I don't want to confuse this, but they're going to receive the information from the sensory neurons, aren't they? They're going to receive that incoming information. They're the ones who are going to, quote, think about it, process it, integrate it. So these are the connectors. I'll just put connect for now. And they were located wholly and completely within the CNS, right? They're completely within the brain and completely within the spinal cord. Now this structural class, I'll mention this in lecture as well. Neurons come in a few different shapes, okay? Let me just introduce that to you right now, and then we'll fill that table in. So neurons come in three different shapes. We can have unipolar, bipolar, and multipolar. Now, when you hear the word pole, what comes to mind? Besides white bears. talked about polarity back in chemistry, and we talked about polarity back in cells. Cells know their top from their bottom, right? They have a polarity. So polar here suggests like a north and a south pole. In other words, a bipolar neuron, here's the cell body. I'll start there. I think it's the easiest to draw. A bipolar neuron, there's the cell body. It's going to have one axon and one dendrite. We're going to keep it really simple. It's bipolar, right? It's got two poles. It has one dendrite and one axon. We won't see very many bipolar neurons this semester. In fact, we won't see any. They're going to be found mostly in your eye. They're a lot involved with your vision. And we won't get to that level of understanding this semester. So we won't label a bipolar neuron this semester. Unipolar, we're going to see a lot of these. Unipolar, there's your soma, there's your cell body. It has a single pole. And then it comes with like a letter T. And one side is the axon, the dendrite on the other side. Do you see why it's called unipolar? It has one pole, if you will, coming from the soma. Your sensory neurons are all going to be unipolar. Okay. So go back to that table, and under class, for sensory, put in the word unipolar. The top right box. It says what structural class do the sensory neurons belong to? They are unipolar, one pole. Then multipolar. What does that suggest? Lots of poles. And what this is going to look like in my basic schematic is you have the soma, and there'll be many, many dendrites and one axon. Okay, multipolar. Many, many dendrites. In fact, we know that some neurons have up to 10,000 dendrites. Everything we draw here will look very simplified. Everything you'll see in your textbook will be very simplified. But in reality, there are some neurons that have 10,000. Some have even suggested as many as 100,000. It's hard to believe that there could be 100,000 little dendrites coming off from a cell, but there's some evidence of that in the cerebellum. So that's a multipolar. Now, multipolar, that's the kind we labeled, isn't it? Do you agree? That the picture I showed you and we labeled had many dendrites and one axon. So the picture we labeled was a multipolar. Most of any axons that your neurons that you're going to label this semester are going to be multipolar. And both your interneurons and your motor neurons are of this class. So on your table, for both interneurons and motor neurons, fill in that they are multipolar. Many dendrites, one axon. You guys know how horribly I draw, right? So I'm at this little gathering with my Sunday school class last week, and we did this silly little activity. I felt like I was at like a woman's, baby shower. It's kind of a fun little game. And I won a contest for the best artist. So that tells you the kind of people I hang with, right? But now, it was one of those little, we had a white paper plate in our head, and we were drawing something on our head that we couldn't see. And I was just really lucky, right? Mine actually looked like what we were supposed to be drawing, whereas everyone else looked like chicken scratch. So amazing. It's the first art award I've ever won, and surely the last. Okay, so let's pull all this together into one picture. I've told you a lot of information. The green is the peripheral nervous system. The blue, the central nervous system. Where do your sensory neurons start? Your afferent neurons start out in the periphery, right, in your skin or around your stomach. That's not your central nervous system either. Here it's showing as a finger. So you have these little dendrites. These are dendrites. Little sensors in your finger. And, oh look, what kind of neuron is this? Look at the shape. Unipolar. See a little T, the cell body coming to a letter T? So there's proof in a pictorial way that our sensory neurons are unipolar. That neuron is going to travel into and go to the spinal cord. We've traveled across the divide. We're now in the central nervous system. And that little shape that looks like this represents a synapse. So that is now the synapse going to the next neuron. Now what's the next neuron in sequence? Number 2, completely contained within the CNS, that is an interneuron. So it's receiving that information from the sensory. It's thinking about it, processing it. If that neuron, I'm simplifying this, but if that neuron thinks that the incoming sensory information says that it should do something, it will then send that signal out, this neuron, and that is the motor neuron. And you see that it's originating in the central nervous system and going back out to the periphery. In this case, it looks like maybe it's going to a skeletal muscle for sure. Maybe the one that's going to move that finger, right? Something is making that finger, maybe you burn that finger or you're itchy or scratchy or something, and as a result, you're going to move that finger. This is a skeletal muscle. What do we know about skeletal muscle? What's one of the characteristics of skeletal muscle? It's striated for sure. How does it work? Is it voluntary or involuntary? Voluntary, okay? So this would be a motor neuron that received signals from the brain. That's not shown here, right? We're really oversimplifying this. But this could also be a smooth muscle or a gland sitting here in response to some sort of stimuli. Okay. So that's a little bit about neurons. Now these neurons are traveling from the outside to the spinal cord, and what none of these pictures are showing is that information, much of it, is going to go up the spinal cord toward your brain. Okay. The information traveling up and down a spinal cord is not called a nerve. It's instead called tracts. We've got some vocabulary to deal with here. Tracts are bundles of axons in the spinal cord and in the brain. We're going to just introduce this idea. If you were a bundle, an ascending bundle of neurons, called the ascending tract, would you be sending sensory or motor information? If you're going up the spinal cord, you would be most likely sending what? What kind of information? Sensory or motor? Sensory, right? So if you're heading up the spinal cord, you're bringing all this information into the spinal cord, it's going up to the brain. If instead you were a collection of neurons coming down, you were a descending tract, you would be sending what kind of information? Motor. Okay. So we won't make this hard. Just ascending and descending tracts going up and down the spinal cord. Another idea. What's a reflex? We all have experienced these. What's a reflex? Reflex. We can all read it, right? It's a quick, automatic, involuntary response. I should also put the word repeatable, right? It's quick, repeatable, involuntary, automatic reaction of a gland or a muscle. Describe to me a reflex that you've experienced. Anybody? So, yeah, when they take a little hammer and they hit the patellar tendon, what happens? A little exaggerated, right? But what happens? Your leg goes out. Is it something you can suppress? Can you stop that from happening? No. So it's automatic if they hit it in the right place. It's involuntary, it's automatic, it's quick, right? All of those things that we've just described. What about another one? What if something flies towards your eye? What do? You blink, right? That's a blink reflex. Do you have control over those? Mm-hmm. Do you have control over your reflexes? Very little. Okay, the CIA assumes that you have very little control over them. This is also the basis of a lie detector test. Right? What are they going to do if they're testing you, if they're checking you out, if they're asking you a historical question or doing a lie detector test? What are they measuring? We've all watched some CIA kind of shows or whatever. What are they looking for?
Heart rate.
Heart rate? Can you control that? Not really. They're looking for your pupil dilation. Can we change that? Nope, that's all automatic. They're looking for your respiration rate. They're looking for how much sweat you're putting out. They'll put a little patch on you and they'll see if you're sweating. Another sign of of lying or distress, right? They're watching for a lot of things. Have you ever watched a luau kind of thing and guys walking on hot coals or something? Now at first, right, that would not have been a fun thing. And if we stepped on a hot coal, what would our body do? We would pull back, right? We would reflexively pull back away from that heat. But it's pretty obvious that you are able to, with a lot of practice, in some ways, perhaps modify or have a little bit of control. Not very much. And like I said, the FBI is basically assuming that you have no control over these reflexes. But we know that lie detector tests are, quote, beatable, which is why they're not permissible. Not everyone responds exactly the same way. So you reach your hand out and you touch the stove. What happens? Even before you were aware that you burned yourself, what did your body already do? It pulled back, right? Reflexively, your hand pulled back. Then you looked at your finger and went, oh, darn, that hurts, right? But you weren't even aware of the pain until after you had pulled away. Or you stub your toe, right? You stub your toe, and immediately, even before you're aware of it, right, you're going to lift that leg, put the other leg down, and then you're going to go, oh, shoot, that hurt. Right? You're not going to be aware of the pain until after your body has already responded. So these reflexes are a protective mechanism. They're saving your eye when you blink. They're saving your hand from burning when you're burning or hurting yourself. What kind of muscles are moving in a reflex? When you pull your arm back, what kind of muscle is being moved? Skeletal muscle, right? And normally we would say that skeletal muscle is voluntary, that you have control over it. But here's one situation where you don't, right? You do not have complete control over your skeletal muscles in a reflex situation. Now, the spinal cord is more than just a connector between the brain and the rest of the world. It actually is in charge of reflexes. I just said that your reflexes are involuntary. That means I don't use any brain power. There isn't enough time. There isn't enough time for that signal to go up your spinal cord, be processed by your brain, and come back down, and then tell you to move. So reflexes don't involve any brain power. They are completely contained and controlled by your spinal cord. So the spinal cord is more than just a connector. It has a job. We've already said that the spinal cord is going through the vertebral foramina. It's about as big around as your pinky. Okay? It has to be, maybe even a little smaller, maybe more like the size of a pencil. Okay? I think we picture something larger. If you look at your vertebra, right, if you think back to any of the vertebra that we saw, right, your spinal cord has to be able to go through that vertebral foramen. Okay? And then there has to be more stuff around it. So it's about as big around as a pencil. And it's going to go down as long as, or down to about L1 in an adult. As we've already said, there are 31 pairs of nerves that are coming off from that, broken up into those regions. So let me now describe that to you. Cervical, thoracic, lumbar, and sacral regions of the spinal cord, even though it only goes down to L1. At around the cervical region and the lumbar region, you're going to see a little bit of a bulge, an enlargement. It's not that obvious, but there's two bulges. It's not just a straight pencil. There's a little bit of bulge up in the cervical region, a little bit of bulge down in the lumbar region. That'll make sense to you why that is as we proceed. The spinal cord itself ends at a structure called the medullary cone. That's the English. In the Latin, it's the conus medullaris. I will accept either one of those words. The conus medullaris or the medullary cone. Sort of like a sharpened pencil. So the spinal cord comes down, and at the end of the sharpened pencil, there is a point, a cone. That is going to be found. The conus medullaris will be at about L1. Everything below that, L2, L3, L4, L5, going down into the sacrum, that is going to be a collection of nerves that resemble a horse's tail or a pony's tail. And for that reason has that terminology from the Latin. Cauda, meaning we know tail, equina, horse. Right? So the horse's tail, the cauda equina. And there I will not accept horse's tail. Right, we got to have the Latin, the cauda equina. So let's take a look at this picture. And we can see that the spinal cord is continuous with the brain. Really what we would call the brain stem area. It passes through the magnum foramen, goes down the vertebral foramina, a little bit of a bulge up here in the cervical region, and then a little bit of a bulge down in the lumbar region. And right here, not the most obvious, but right there, the spinal cord ends at the medullary cone. Comes to a kind of a sharpened point. Everything below that collectively is referred to as the cauda equina. The cauda equina. Now, coming off from the spinal cord, we see the spinal nerves, right? There's the 8 cervical nerves, and then there are the 12 thoracic nerves. There are 5 lumbar nerves, 5 sacral nerves, and this little dude right there is the one and only coccygeal nerve, CO1. The spinal cord is important. Again, it doesn't regenerate, neither do the neurons of the brain very well, and so there are some layers around the spinal cord. These are collectively called the meninges. Where have you heard the term meninges or meningitis? What is meningitis? It must be itis, inflammation of this stuff, right, the meninges. So meningitis would be an infection in these protective coverings around both the brain and the spinal cord. So the meninges, here I'm describing the meninges of the spinal cord. It's largely the same for the meninges of the brain. Okay, it's the same 3 layers. The 3 layers, the dura mater. What does dur mean from vocab? Dur, hard, or think it's durable, right? The durable, it comes all from the same word from hard. So this is a tough collagenous, remember collagen, think strength. This is a tough outer layer that is going to protect the entire brain and the spinal cord. It is surrounded superficially by the epidural space. Where have you heard of the term epidural? During pregnancy, oftentimes, not during pregnancy, but during labor, right? Oftentimes, a woman will receive an epidural. What's the epidural? What's it supposed to do? Numb from the uterus down, right? And everything from that point down is numb during the pain of childbirth. We'll look at where that epidural space is in a moment. Then there's the arachnoid mater, or simply sometimes called the arachnoid. What does it suggest its shape is like? What's an arachnid? Spider, arachnoid. This is a spider web shaped area. It's kind of got some fibers. It kind of looks like a spider web. And it's underneath this arachnoid mater that there is CSF. CSF is cerebrospinal fluid. Spinal, sorry. That's an O. Cerebrospinal, all one word. Cerebrospinal fluid. And that CSF is going to be around the brain as well. And then finally there is a very delicate layer. I think of it as being shrink-wrapped right to the surface of the brain called the pia mater. And the pia mater is going to follow every groove and bump along the surface of the brain and along the surface of the spinal cord. And we're going to see that coming off from the pia is a structure called the filum terminale. And let me go back and show you that filum terminale. It is in this picture, this red, it is in this picture, this red structure right here, right down the middle. Okay? And that is the filum, or the terminal filum, or the filum terminale. And that is an extension of the pia, and it anchors the spinal cord down to the coccyx. You'll see that on the models as well. So let's take a look. In the past, well, first of all, what kind of vertebra is this? You've seen this vertebra before. You've never seen it filled in with the spinal cord before. What kind of vertebra is this? Cervical, because it says so, or because you know from looking at it? What? Yeah, the transverse foramina, right? See the 2 extra openings? Okay, so we see the two extra openings, so we remind ourselves that is a cervical vertebra. Now, what do we see here? We're used to seeing a big open hole. Now, I see that hole is filled. The spinal cord itself is very distinctive. It has what is described as being sort of a butterfly-shaped core, or a letter H. And we can see that letter H, or that butterfly shape, in the brown here. So, and then the spinal cord itself is around it. You'll notice that the butterfly itself is a dark color. That would be called the gray matter. And around it is the white matter. And we'll talk a little about white and gray matter. In the very center of it, there's a canal. That central canal, just like the name used in the osteon, but a very different structure, the central canal is where one finds more of that CSF, that cerebrospinal fluid. So you have fluid not only around your spinal cord, but going right down the center of your spinal cord. The same is true in your brain. You've got fluid around your brain and inside your brain in some structures we'll discover later. The layers here, the salmon-colored, orangey color, that is the dura. Okay, it looks kind of tough, kind of a tough layer. Right above it, in this fatty-filled area, that is the epidural space. That's where the nurse anesthetist or the anesthesiologist is aiming when they're trying to give an epidural. They're leaving that anesthetic drip in that little epidural space, and that will numb the nerves from that point and everything down. The lighter blue is the arachnoid mater, and you can see those little fibers. Those little fibers are the spider web appearance. The darker blue is a fluid-filled space called the subarachnoid space, and that subarachnoid space is filled with CSF. Okay, so that's where the CSF is, in the subarachnoid space. And then the pink, that is shrink-wrapped right to the surface of the spinal cord itself, the pink is the pia. Coming off the spinal cord, what do you see? I see some yellow things. What are those? Those are the spinal nerves, right? So here's a spinal nerve coming out over here. Here's a spinal nerve coming out over here. That's all we see. On our models, all we see is a single structure coming out, right? The spinal nerve. What we don't see is that deep inside the vertebra, that spinal nerve splits. And it's going to have a front part and a back part. Let's get our orientation here. There's the spinous process. So I know that is what direction? This is posterior, right, or dorsal. That makes this down here anterior or ventral. Look at the butterfly wings. You can always get your bearings. If you look, if the vertebra wasn't here and you didn't have the spinous process to look at, or the body to look at to get your orientation, if you look at the wings of the butterfly, the wings always reach and essentially touch on the dorsal side. Notice the wings are not touching on the ventral side. Okay? So you'll always be able to get your orientation by looking at the spinal cord. The tips of the wings are always pointing toward and touching the back side. Here's the same colors, the same, everything's labeled, except now we're seeing the spinal cord in a more of a three-dimensional column. And I like this picture because now you get more of an appreciation of what's going on. Same structures. The orangey layer is the dura, the light blue is the arachnoid, and the pink is the pia. There's a lot more to talk about. We'll get to a lot more of this detail in lecture. I just want to get us through enough. I'm giving you enough as it is. There's white matter and gray matter. I told you that there was a cell around the axon. What was that cell? Schwann cells. And what did that Schwann cell make? Myelin. And where did we find Schwann cells? Only wrapped around what part of the neuron? Axon. Okay. And the myelin is white. So wherever you see white matter, you know you have axons that are wrapped with myelin. Wherever you see gray matter, it's not white. So what isn't there? Gray matter would not be surrounded by what? Myelin. What parts of the neuron, think back to the picture, what parts of the neuron did not have Schwann cells around it? You did not see Schwann cells around dendrites. You did not see Schwann cells around the soma. So wherever you see gray matter, it is essentially non-myelinated structures. Wherever you see white matter, that's where you likely have mostly axons. Just a couple more ideas, guys, and then we'll break out into some sections, and we'll be looking at some things today. Here's the spinal cord. What do we have? Let me point to a couple things. What's in the very center of that spinal cord? The central canal, good. And what's in that central canal? CSF, cerebrospinal fluid. And we see the gray butterfly. And then the white matter around it, here the diagram is trying to show you that the white matter is broken up into areas called tracts. We've already mentioned those. The ascending and descending tracts. What do the ascending tracts do? The red areas would be sending information up the spinal cord to the brain, and the green areas would be sending information down, motor information down. Now they're on both sides. They're leaving out the opposite colors, but all through that white matter around the gray butterfly, you're going to have axons traveling up and down in these bundles called tracts. Let's talk about reflexes for a moment. This is a pretty cool thing. We've all experienced this. In about 3 months, hopefully we can be outside with some bare feet on. And this guy is going to step on some glass. Now, he stepped on glass with his right foot, and it hurts, right? But even before he was aware that he was bleeding or that he had pain, what happened? It's hard to see, but there's a yellow sensory neuron. There's a little yellow arrow right here. So his sensory neurons in his feet sensed that pain, and that signal went up and went into his spinal cord. Oh, look, what's that thing? That little pinhead tells us that that sensory neuron is a unipolar neuron. It comes in, it always comes in on the dorsal side of the spinal cord. Now, what is that? Remember, we don't have time for the information to go to the brain. This is the reflex. So it's going to come in and synapse, talk to an interneuron. The interneurons are shown in purple. And then that interneuron is going to think about it and say, we got to do something here. And it's going to connect to a motor neuron. The motor neuron always leaves out the front side, the ventral side of the spinal cord, and goes back down. Now there's actually 2 signals going down. One is going to his right leg. And it's going down to his hamstrings. What are the hamstrings doing? Flexing, right? Lifting the leg away from the pain. But you also see a signal coming down and going to his quadriceps. What would that signal be telling the quadriceps to do? It's not so much that it's telling it to do anything, but it's going to inhibit it and tell it to relax. Allow the hamstrings to do their job. Remember, muscles are extensible, and they have to work against the antagonistic muscle group. Now, when you lift up your right foot away from that pain, you don't fall down, do you? What happens? As part of that reflex, your left leg is going to extend. And your weight's going to go over to your left side, isn't it? This all happens faster than you can even imagine it. So that means that there has to be signals going across to the other side of the spinal cord. And now motor neurons, efferent neurons, sending signals down to the hamstrings and the quadriceps. What are they telling the quadriceps to do? Right? To extend, right? So they'd be telling the quadriceps to contract and telling the hamstrings to do what? Relax, right, if you will. Right? See what's going on? We talked about this antagonistic group, the hamstrings and the quadriceps. That's my 8th or 9th or 10th lie of the day, right? You have to remember some of the stuff from previous semester. This is all happening so fast. There's no brain power involved at all. This is all happening at the level of the spinal cord, and this is a reflex. It's called a polysynaptic reflex. Why poly? Poly means many, right? There are multiple synapses involved. You agree? I mean, we've got to come in and talk to a bunch of interneurons and a bunch of motor neurons. There are a lot of neurons involved. That makes this reflex a little bit slower than some other reflexes, but pretty complex in that even before we were aware of the pain, our other leg has extended and we hobble away from the pain, but we've saved our foot, right? Hopefully we haven't had any extra pain or damage. Okay, and this is just the reflex arc, and then I'm going to let you guys head off to the stations. There are 5 steps to most every reflex arc. This cartoon shows those to us. Number 1, there's a receptor. Here it's in your skin. There's a receptor in your skin. And what did that receptor notice? Ouch, right? Pin comes in, something pokes you. That sensory neuron sends it in. Oh man, there it is again. What's that little push pin tell us? That sensory neuron is a unipolar neuron. It comes in, talks to an interneuron, and then talks to an outgoing motor neuron, and looks like it's going off to some sort of muscle, doesn't it? So those are the 5 steps. There's going to be a receptor. There's going to be a sensory neuron that's going to carry that information to an interneuron. The interneuron is going to send that information now out of motor neuron to the effector. Effector is either muscle or gland. In this case, it looks like a muscle. The effector is the target, okay, the muscle or the gland. As we go through the stations today, we're going to be looking at the brain, we're going to be looking at the eye, the ear, the spinal cord. There's a lot of stuff today, and it's not all going to make sense to you. Today is one of those see it, find it in your book, start making lists, marking up your pages, and then as we progress through this information, learning about the neuron, learning about the brain and the spinal cord, then you'll be coming back and reviewing this information, and it will all be restated again in lecture, and it will all start making sense to you. The eye and the ear are also part of today's lab. The next test won't even include the eye and the ear. The eye and the ear are coming even further in the semester. So actually this lab is covering about 3.5 weeks of lecture material. So you're being exposed to it today, and then you'll be reviewing it and looking back at these lists over the next couple of days. Let me just start off with the brain. Let me grab your brain over here, Leah. Now the brain, I just want to introduce a couple things. This is now the 15th lie, because the brain is divided up into lobes, sections. And you already know the names of these lobes, because the bone that protects that lobe has the same name. How many frontal bones are there? 1, right? But that frontal bone protects both the left and the right frontal lobes, these sections of your brain. How many parietal bones? 2, they protect the parietal lobes. We got this. Temporal, 2 lobes, 2 bones. Occipital, 1 bone protecting both the left and right occipital lobe. Your brain is magnificent. When we get to the cardiovascular system, we'll talk about the heart, right? It's 2 pumps sitting side by side. The brain is much the same way. Neurologists will talk about the left brain and the right brain. They are 2 organs, essentially, talking to each other, connected intimately together. But they do have their own features, and they're not mirror images. So your left and your right heart are different, your left and your right brain are different. And we'll talk about some of those differences in lecture. But for now, you'll be looking at some different structures of the brain. Let's think about this, though. The frontal lobe. I'll get through this and then I'll shut up for a while. The frontal lobe. The frontal lobe is responsible for motor movement. This is the front. It's the front of your brain that is telling you to move. So when you decide to move, that information is coming from your frontal lobe. This is also the part of your brain that helps you make really good decisions. So executive function and thinking through a process, much of that comes from the frontal lobe. So that means that if a person had damage to their frontal lobe, they're going to have perhaps problems with movement. They can't initiate the movement. They may have some paralysis, or they may start acting very strangely. This is the area of inhibitions. So if someone has a frontal lobe damage, they may start swearing like a trooper. Right? Grandma may start swearing like a trooper. Or Uncle John may start doing some really strange things he never would have done before he had that brain damage. Again, this is your areas of inhibition, your thinking, and your voluntary movement. Parietal lobe. This is the area that's receiving information. So if you think about it, the spinal cord goes straight up, right? You're ending up right at the parietal lobe. So the parietal lobe is receiving sensory information. If you had damage to the parietal lobe, you may not be able to understand your environment. You may not respond well to the stimuli that are coming in. Temporal lobes by the ears. There's a lot of hearing centers there. That kind of makes sense. We'll talk about that for now. The occipital lobe. Strangely enough, the occipital lobe is or has within it your vision centers. It's kind of far away from your eyes, right? But the occipital lobe is where your visual centers are. So the information's coming in your eye, and not all of it, but much of it's going back to your occipital lobe where you sense and understand vision. I remember that one. My grandmother, when my mother was 5, so this is about 1950, my grandmother was ice skating and hit the back of her head very, very hard in a fall. And within 5 days, she was completely blind. Now, they never really understood what happened. Now we know what happened, but they couldn't do anything back in 1950. We didn't have MRIs and CTs and that kind of technology. Likely what happened is that she hit her head, had a bleed, right, and damaged a part of that occipital lobe on both sides, and so she lost her vision in both eyes. Nowadays, if that had happened, the person we rushed off probably had a headache, we rushed off to the hospital, maybe could have stopped that bleed and maybe saved her vision, but we didn't have that technology back then. So that's what I remember. If you hit the back of your head, you see stars, that kind of idea, absolutely you have interfered with your visual centers. So that's a lot of information, folks, and I understand how overwhelmed you're feeling right now. I'm not going to go over the eye and the ear. You'll look at that yourself right now. Let's just go to this last page. Each of you should find at your table a laminated page that looks a lot like this slide. And this is our road map. This is our road map, okay? Each of you should have a different red box circled on your particular table. And what that tells you is what pages, and what section of the Amerman book you're heading to. So if you were at, for example, table 1, okay, and that's this table up here, table 1, and they should have a red box around table 1, and it tells them that they are going to head in their Amerman book to 13-1, pages 283 and 288. Go ahead and let's everyone do that right now. Everyone go to your Amerman book, pretend you're at table 1, go to 13-1, this is about the brain, and look at those pages from 283 to 288. Toward the end of that material, right, 283 to 288. Are my pages off?
Oh, yeah, your chart, it says 307 on the thing.
Okay, my numbers are off. I have the wrong semester up. I apologize. On the page, it's right, though?
Yeah.
Bad me. Okay, old edition. So page 307 to page 312. I apologize. So forget what's on the screen. Now, in that group of pages, it starts off with an introduction of the brain, and go toward the end of those pages, go back toward page 312, and on page 311, you see a list of structures called the model inventory. This is your list of structures that you are to identify on the brain. You're going to find a similar list in each of these sections. There'll be a list for the spinal cord. There'll be a list for the eye. There'll be a list for the brain. There'll be a list for the ear. On your pre-lab, you should have already crossed off some things. It told you to omit the following structures. If you did not do that, I will quickly evaluate your pre-labs and get those back to you so you know which things to ignore, to cross off. Okay? So that's your job. Each table should have, or I will put out, your key. So this model key in this notebook are the manufacturer keys for all of these models. And if you'll turn to the nervous system section in here, you will find a key for the brain or the eye or the ear or the spinal cord model. We have about an hour and a half left. So that means that we have about 30 minutes per double station, about 15 minutes per station. That's not enough time to master it, but it is enough time to at least find everything and to mark it in your book and then start creating that study guide. So tables 1 and 2, about 15 minutes each. After about 15 minutes, I'll remind you it's time to switch. You can physically move, or you can just simply move the models to the other side. So you're going to have about a half hour to look at the brain and the spinal cord and the ear. On the far side over here, tables 5 and 6. We've got a diorama of a neuron, and there's a key for this. There's a poster here of nervous tissue, and there are two microscopes set up. The microscope on this table are some giant neurons that are under the microscope. Then at the other table you have the spinal cord. There's a three-dimensional model of the spinal cord plus spinal cord under the microscope. Again, about 15 minutes per station. Check off structures, start to identify as many as you can. Then in the middle table, this is going to be our dissection table. And here you're going to start off with dissections, and then other groups will come here as well. Again, about 15 minutes per table. We have eyeballs. The eyeballs will be done in groups of 2, or if there's a group of 3 at your table, you can do it as a group of 3, or if you want to go out on it at your own, you can. The instructions for this are found on the pages. So the eye dissections you'll find in the pig book, page 105 to 108, or Amerman, 339 to 343, and those numbers aren't right, right? I'm going back to the wrong numbers. But on your table, Piggy page 105 is right, and then the eye dissection page 367 in your Amerman, and then the other table has some brains at it. To make this more simple, I'll have both of you starting at the same thing. So I'm going to have you start with the eye, and then we'll all switch over to the brain. In your supplement, you're going to find a checklist for dissection rubrics. You are to put your, before you do anything with your gloves, go ahead and put your name on this for your team. I only need one of these per team, and put both your names on that. And then today, this is practice. This is practice. There's no quiz grade today. But normally this dissection rubric will become the same as that post-lab assessment. Rather than having 5 questions at the end of every lab, from this point on we'll be doing a dissection almost every week. And if you get all the structures correct on this dissection rubric, you'll get those points added to your quiz grade. I will come by and look at this. At your table, I'm losing track, I'm losing control. You should find a little container of pins. Jennifer has spent hours, literally sometimes, trying to make sure that each of these has a 1 through 20. Please try not to mix your pins up into different groups. It just gets frustrating. So try to keep your groups separate. You're going to be labeling, for example, if you're over at the eye station, on the eye dissection rubric, it tells you the sclera is number 1. So put pin number 1 in the sclera. Number 2 is the iris. So put pin number 2 in the iris to the best that you can. And then I'll come by and I'll check that, and I'll check you off before you're done with that. So we all have a little something different to do. I'm here to get you started. Let's go ahead and, if you're in the middle, grab your lab coats and your goggles and your gloves. The rest of you can start looking at your particular models.